Before I begin, I want to thank the organizers for this opportunity to come and talk at this summit, which I was particularly excited about because a lot of these antifungal agents also have activity against malaria, and for us, one of our model systems is yeast and fungi. So for what I'm going to be talking about, the discovery of molecules to probe and treat Plasmodium, I hope that it's not something that was too different from some of these other exciting talks. And I think that this audience doesn't need too much of a background on malaria, but I always like to show the parasites that we're trying to kill from Plasmodium, shown here. And also the map where you can see the distribution of malaria cases across the globe. And in particular, you can see that there's problems in Africa, Asia, and South America. I think there's been some great work in the past few years by the World Health Organization and private companies like Bill and Melinda Gates to reduce the number of malaria rates. And in particular, you can see now we're at around 300 million cases per year and only 600,000 deaths. And so this is a great feat. But unfortunately, all of the prediction is indicating that this is going to be slowing down, if not rising again. And in particular, this is from parasite drug resistance that's come up. And now with the Ebola outbreak in Africa, a lot of the countries that are suffering the most from this disease are not getting medications. So we are thinking there's going to be a huge rise in the next couple of years. To address this, we really need to have either a vaccine therapy or new drugs that can work on these parasites that are developing resistance. But unfortunately, once things go out into the market, they can develop this resistance or loss of sensitivity quite rapidly. So our goal is to try and identify things with new targets. And one way I wanted to tackle this was really to look at the life cycle of the parasite to see how we could do this best. And this life cycle will start with an older mosquito, shown here, taking a blood meal. And when this happens, she can inject anywhere from 10 to 30 parasites into the skin. At this stage, these parasites will travel side by side with red blood cells, completely ignoring them because they have the sole purpose of finding the liver. And it's in this liver that that one parasite will enter and exit several cells as a process called traversal until it finds one that it will replicate in. And then that one parasite becomes 10,000 in just a few short days. This is completely asymptomatic, but once these parasites leave the liver, they no longer invade liver cells, but they can invade red blood cells. And this starts the cyclical invasion and bursting of red blood cells that will lead to malaria's characteristic symptoms, the fevers and chills, and also put people at risk of death. It is also during this red blood cell stage that a form of the parasite breaks off that can be picked up by a new young mosquito. And so this is how this one organism can cleverly jump between a mosquito and a human, and within the human, between the liver and red blood cells. To date, almost all drug discovery campaigns are focused on the cyclical red blood cell stage. And this is very important because this is when people are sick. But you could imagine if you also had a compound with activity against this liver stage, you now don't have a treatment strategy, but a prophylactic one. Additionally, at this stage, you're dealing with a much smaller number of parasites, around 30, as opposed to around 1 billion, which can be in the blood stage. So this means that it's an easier number to kill, but also they'll be less likely to develop drug resistance. And this was really hard to do because we didn't have the models efficiently to have this liver stage in culture, so this is a very elusive stage. We know little about its biology. But just recently, we've been able to do high-throughput screening on that particular stage. There are many reasons I think that you could identify things that have either liver stage activity or blood stage activity or potentially both. And this is just a Venn diagram showing proteins that were isolated either during the blood stage, liver stage, or both. So you can see that there's quite a bit of target space in either one of these parasite forms. Additionally, there are known molecules that on the market that can clear blood stage parasites but don't have activity against liver stage parasites. We know of primaquine, which has activity against blood and liver stage parasites in vitro, but just a couple years ago there was no molecules that had just liver stage selective activity and just one or two that had this dual stage activity. That's because we didn't know how to look. So when I was a postdoc at Harvard Medical School in John Clardy's lab, what I did there was develop a high-throughput screen, shown here, to assess molecules for activity against the liver stage of malaria. And this is done by first plating Hep G2 cells, a liver cancer cell line, in 384 well plates, and adding drugs to this. And then after that, taking live mosquitoes and dissecting out parasites from their salivary glands. Once that happens, we can use the parasites in the salivary glands to infect the liver cells, and in 2 days either measure a parasite load which we can do because we have a luciferase reporter on our parasite strain, or measure just cell viability, because we want compounds that inhibit this parasite load but have no activity against liver cell viability. And I should also say that instead of using a human malaria strain, which would be BSL-3, we're using a Burkii or a mouse malaria strain. And this is useful for us because that means we can also jump into mouse studies once we find things that are active. What I was particularly interested in were compounds called known bioactives. So these had previously reported activities, some of them FDA-approved drugs or things in NIH clinical trials, but we didn't know if they would have activity against malaria's liver stage. Because there was this previously reported biological activity, we could jump off of these previous efforts to form a hypothesis about potential targets in liver stage malaria. And I should say that to date there's been no successful work done that has a target of a small molecule against the liver stage parasites. So by first testing a small subset of around 5,000 compounds of these known bioactives, we found that there were quite a bit of actives on the primary screen, so 70. And these were compounds that inhibited the parasites 95% or greater at 10 micromolar. These were retested from a plate and then purchased to see if they would also retest. So we had 55 retesting when we repurchased them. And then when we did IC50 analysis, we found that we had 37 compounds that had an IC50 of 10 micromolar or below. So we called these our leads. Also, these did not have activity against the liver cells. And what was very interesting to us is to take the hypothesis that we could form from these structures and group them into either antiparasitic agents, anti-cancer, antibacterial, antifungal, or what I'm just going to call other because it didn't group into any of these other three categories. And what you could see, as I said earlier, that a lot of these antifungal agents also had activity against malaria, but also we were finding things that in this other category, which we have no idea how or why they're working against liver stage parasites. So this is particularly exciting for us. Additionally, we were able to pull out from this library primaquine, which was the known liver stage malaria inhibitor. So this was a great validation for Assay. But you can see that primaquine actually had the highest IC50 among all of these compounds that we identified. So it didn't set a very high bar for us. But to be fair, our lowest IC50, or our most potent compound, was atovaquone, which was the other molecule that we know of that has this dual stage activity against liver and blood stages. But besides that, we had very diverse chemical structures, as demonstrated here, some of them over-the-counter drugs like Nexium. This was particularly exciting for us, and we had the question of what are these molecules doing to the parasites to cause this inhibition. So as a chemist, what we can do is take the structures of these and then hit them in a database and inform potential targets with chemical informatics. And that's where we're able to get this pie chart of predicted targets shown here. So the majority of our compounds were targeting ligand-regulated receptors, and this receptor would be anything from something that would bind estrogen, histamine, or serotonin. Also, as a major group, ionoforms, or things that affected metal regulation. We had some known antimalaria targets like dihydrofolate reductase and HSP90, but for the most part, we have a lot of diverse potential targets that our lab is actively pursuing to see if these can be validated. And so just for the purpose of this talk, I'm going to go into a little bit of the work with the kinases that we found. Because these were both blood and liver stage inhibitors, so they have the potential of both being treatment and a prophylactic. But one of the very, I think, interesting yet challenging parts of my assay is I'm telling you, we pick compounds because they have a known biological activity. And I'm dealing with a parasite, it's Plasmodium parasite, that resides within a hepatocyte. So we have the question of whether or not these small molecules are influencing parasite load by affecting the hepatocyte itself, the known target, or the Plasmodium parasite. And to tease this apart, we have two main strategies. One was an RNAi interference strategy, so knocking down genes in the host that we predicted were the targets of these small molecules. And we can't use the same strategy in the parasite because it doesn't utilize microRNA as a means of regulation. And I can say right now that we've done this for every predicted target and have yet to found effect. So that suggests to us that the small molecules are not influencing the host by its known target. So then the next kind of easiest path for us is to do an activity profiling study. So looking around our active molecules and doing SAR to see if we can find a pattern that's different between the parasite and that in the host. And so one molecule that we did this with was this CDK inhibitor shown here, and it's aptly named CDK12 because it inhibits the 1 and 2 isoforms of the cyclin-dependent kinase in humans. And again, this is a dual-stage inhibitor, and in both assays, blood and liver, it was around a 500 nanomolar inhibitor. So just by purchasing other small molecules that also target human cyclin-dependent kinases, we could get quite a range of activities and potency, which is indicated with the X here. But if it's in gray, it means that it did not inhibit neither our blood or liver stage assay. But if it's in white, it did inhibit, and I just have the liver stage IC50 shown here. And from this small subset, we were able to identify small molecules that were more potent at inhibiting liver stage parasites, like AT7519. But we wouldn't have predicted that because they're actually less potent at inhibiting these human CDK12 isoforms. So this seemed to be another validation that the target that we're looking at is not going to be a human kinase, but in fact a parasite one. So we were able to build off some of the great work done in Oliver Bilker's lab in England, where he did all of the Plasmodium berghii kinases, which are represented in this kinase tree, and systematically did knockouts. And from that, with these 66 predicted kinases, he found that 43 were likely essential because if you knock them out, you weren't able to get viable cultures. So this was an absence of a phenotype, and what we were interested in doing is saying, okay, now that we think that kinases are very important for malaria, can we now use our screening strategy, instead of doing this unbiased library, but a more focused screen just on kinase inhibitors, to see if we can kind of replicate which ones he's predicting to be essential. So we worked with GlaxoSmithKline, Roche, Novartis, and some other kinase experts at Harvard like Nathaniel Gray, and got a library of around 3,000 small molecule kinase inhibitors and tested them in our liver stage malaria screen. And based on the known activities of those compounds, we found all of the genes highlighted in yellow are likely going to be essential for malaria. So that is, these were being targeted by multiple chemotypes that were predicted to hit homologs in the human. And in particular, we were interested in this PK5. So this is a member of the cyclin-dependent kinase family in Plasmodium. And the largest number of our hits were in fact cyclin-dependent kinase inhibitors. So on this, you can see in red where the Plasmodium PK5 lies, and its closest homologs in human, which are in black, which is CDK2 and CDK1, and in blue is yeast. So there are other cyclin-dependent kinases in malaria, shown in red here, but they are not as homologous. So this led us to believe that the target of our CDK inhibitors is in fact PFPK5 and not the human kinase in the system. Unfortunately, there was a crystal structure of PFPK5 by Jane Encat's lab in England. And here's the structure, but what we did is work with Peter Slitz's lab at Harvard to dock in all of our small molecules to see if they were predicted to bind based on their binding energies. And in fact, all of them were predicted to bind. So everything that was a cyclin-dependent kinase inhibitor was a hit in our screen. We predicted it to interact with the protein. And then we were actually able to do biochemistry experiments to show that it did in fact bind, and in this case with a KD of 330 nanomolar shown here. So this seemed to suggest that this was going to be an important kinase in the parasite, and we were able to take our same mosquitoes and go into a mouse model shown here. So in these mice, they were treated with 30 milligrams per kilogram of a drug 3 hours before dosing with the parasites. Then they got 1 dose of the parasites, and this is their liver 3 days later. And you can see with just that 1 dosing of the inhibitor, here you were able to reduce that parasite load. So I think that these molecules are working both in an in vitro and an in vivo setting. But the main challenge, which I think was highlighted in the previous talk, is this issue of potency and then selectivity. So we identified these compounds because they had activity against human CDK2. And what you can see in blue, for each one of these compounds, they were more active against the human isoform than the parasite in white. And this is further demonstrated on this plot here, where you can see the IC50s in the nanomolar range for the human isoform, and then for the liver stage IC50, it's micromolar. So we're dealing with tenfold selectivity, but in the wrong direction. So what we wanted to see is, can we engineer a new molecule that now has potency in what we want to call the right direction? So for PFPK5, but not human CDK2. And this is where we decided to do something not like what we had been doing previously with these phenotypic assays, But what I'm going to say, take a slightly easier route, which doesn't involve the decapitation of 10,000 mosquitoes. So in this in silico approach shown here, we were able to just take molecules from a accessible compound library and then screen them for potential interaction with PFPK5. We could do a counter screen with the human CDK2 and identify our top hits, and then try and do biochemistry to show if they really do bind, and if they do, if we can improve it. And you say that this computational work was all done in collaboration with Peter Sliz's lab at Harvard. And Marie Perkins and Amber Harold in my lab has been doing a lot of the biochemical studies. And so this is where, previously I had a number like 5,000, and now I can take a number like 350,000. Again, a very diverse library. They screened it first with standard precision and then extra precision, and then they did 2D fingerprinting to get kind of unique chemotypes. And we took the 184 of the top hits, and we did a primary screen against both the Plasmodium PK5 and the human CDK2. This was at 8 micromolar, so a one test. And then we would do KD determinations to see how selective they are, and a secondary screen. And I should point out that the primary screen, the reason we picked these accessible libraries is I could get them through a cherry pick with a screening facility, and then we could repurchase them for the secondary screen. So what we found from those 184 compounds, that if we just did them at that one concentration of around 8 micromolar, a lot of them would bind to the human CDK2 and the PFPK5. And there wasn't a ton of selectivity, so these are around a 1-to-1 ratio. We did find a few that were selective in the opposite region, but what we were really excited about was this group of 3 here, which seemed to now have selectivity for the Parasite PFPK5 over the human CDK2. And that's shown here with the percent bound just at that 8 micromolar. And so for all of these, again, we had a cherry pick plate, so we purchased these and tested them with a KD analysis, which is shown here. And what we found is 2 of the compounds didn't repeat when we purchased, but we did have one molecule shown here. Oh, sorry, here's the molecule and the data that did bind to both PFPK5 and human CDK2. And what we were really excited about is now we even had some selectivity. And this is a reproducible 2-fold selectivity for binding to the parasite cyclin-dependent kinase over the human one. And this will be the first time that we've ever had selectivity in this direction. Because if you remember, in fact, we had 10,000-fold in the opposite direction from our previous strategy. And when we tested this molecule against liver stage and blood stage parasites, we see it also has activity in our in vitro models. So we have a compound now that we think has a nice predicted target, which is this parasite PFPK5, and that this will have activity in our in vitro and our in vivo assays. So just to summarize, I think that we were able to use this screening strategy first with this wide known bioactive compound library set, and then a more specific one to identify potentially essential proteins. And again, there's been no target identification strategy in this liver stage yet, so that's also an active part for the other molecules we found. But we were able to use one to find PFPK5 as a target, and then do an in silico strategy to identify other potential inhibitors. And we have for the first time something with this mild selectivity. And so what we're excited about now is doing the optimization around this to try and improve that even more. And also our collaborators at Harvard are working to co-crystallize that small molecule with our enzyme. So I just wanted to thank at Harvard John Clardy, who was my postdoctoral mentor, where I did the initial known bioactive screening. I worked with Nathaniel Gray on the kinase inhibitors, and Piotr Sliz's lab did all the computational work. Additionally, Maria Moto at the University of Lisbon in Portugal is another liver stage malaria expert, and they did all of the mouse model work. The NYU insectary is where we've been getting our mosquitoes, and the screening facility at Harvard is where we were able to get our cherry pick plate, and the NIH for funding. And lastly, I'm very fortunate to have a really great lab at Harvard. And in particular, I talked about a project that Marie Perkins and Amber Harold in the chemistry department have been working on. And thank you for your attention.